So this is the last thing we'll cover in the workshop. There is a session 5, which we won't cover, but I encourage anyone who's interested in differential isoform analysis or differential transcript utilization analysis. We do cover that in that session, and it's pretty self-explanatory. It's actually a little bit shorter than some of the sessions we've been doing, and we do have videos of it on our YouTube as well. So if you want to hear me talking about it, or hear one of my colleagues who's also taken this to taught these sessions, you can go and listen to that as well. And so it is really great, but it varies how much people are invested in isoform analysis. But we've seen lots of things pop up in that kind of analysis where we didn't necessarily expect it. So it's often an interesting supplement to your data set. So we're going to switch now back to using Christina Leslie's data. So this is the Treg data and the activated Treg. So we have naive and activated T cells. And we need to be in the same place as usual within this, but also one level down within our course. Everyone should be there by now. And the only thing we really need for this analysis is the results of a differential gene expression analysis. Okay, so group activated minus resting. And so what we're going to cover in this session is really, like, what we've done so far is finding experimentally interesting genes. We define them in various ways, whether they're significantly changing or they drop out from clustering. But functionally, it's not necessarily like you want to actually know what they're functionally doing, these genes. And so you can then start to, by doing enrichment analyses, start to understand, like, is there a gene set that's enriched within my condition? So like, are cell cycle changes enriched when you do this analysis, or are genes related to like immune response or something like that? Is that in one of the clusters versus a different cluster? So that's kind of what we're getting at with this. And so there's lots of different kinds of gene sets, and there's lots of kinds of gene set analysis. And so this can sometimes, people get a little overwhelmed by this, because quite often the way people do GoTerm analysis is they'll just go to some web browser, and then you can throw in some gene sets, and then it gives you a result, right? And you're not entirely sure. When they come and talk to us, they say, oh, I got this result. Can you like double check it? And I'm like, well, what did you do? Did you know what gene set you looked at? Do you know how it was tested? And people like shrug their shoulders. Okay. And so being able to control and do it yourself through something like R is very, very useful, because then you know exactly what you're testing. And so to get at some of the terms associated with this, we have a gene set. And that's basically a named collection of genes. This could be a GoTerm. So this could be like immune-related genes, heart development genes. It could also be like a KEGG term. It could be something from MSigDB, which we'll cover. And so we'll dig into different kinds of gene sets. We also have a gene set lowercase e enrichment analysis. And this is kind of a very broad term for anything where you're looking for functional enrichment, okay? By correlating a set of genes. It's quite often uses as like a statistical test, like a Fisher test or something like that. Then you have gene set enrichment analysis with a capital E. And so this is a technique developed by the Broad Institute, and it's a methodology for correlating a set of genes with a condition or a phenotype, okay? And we'll cover both of these, and both of them are very useful, but they are slightly different. And so starting with the first of these points, it's the gene set. There's lots of different databases out there for this. People often lean straight towards GoTerms. So for the Go consortium, there's also things like the Reactome, which helps you get to biological pathways. I think I mentioned KEGG as well. And then there's MSigDB. And then beyond that, like you can also use anything that you define as a gene set, but you need to have some reason why. You can't just say, these are my favorite genes. You need to experimentally derive them, or say these were from a paper, and you can then reference the paper, and that's where the gene set was derived. And so if we focus on gene ontology, the Gene Ontology Consortium is a very comprehensive resource. It has a huge amount of lists. It has a lot of history. And there's three main categories. There's molecular function. So that's what is the activity of the gene or protein product. Biological process, what is the role of that gene or protein product? And I'd say that's the one people focus on most of the time. And then cellular component, where in the cell is it found? And that's the one maybe least used, but actually it can be super interesting to look at as well. And so the way that Go is organized, Go terms, is this kind of nested structured graph. And so you have something at the top, which is the biggest term, a biological process. And then underneath that, you have regulation of biological process. And then underneath that, you have negative regulation or positive regulation of biological process. And it keeps going deeper and deeper and splitting and splitting. And so some of these terms at the top will be massive. They'll include half the genes in the genome. But they're also not very interesting because they're not specific. But as you get deeper and deeper, you might find you get something very specific, like positive regulation of cuticle pigmentation. Right? And then that is something incredibly specific and incredibly targeted. So if you are able to get that as a result from your analysis, then that is super instructive into what is changing. The reactome and KEGG contains slightly different information, where it's more about genes and their membership in different molecular pathways. So this is really focused on like metabolics and specific pathways that are happening. Often there's a clinical angle as well. And so like it might have like disease pathways. And so these can, depending on your field of study, KEGG and the reactome might actually be more useful for your analysis. And then last, but definitely not least, we're actually gonna, I think we focus a lot more on MSigDB than maybe the others, because it's maybe the least well known. And this is the molecular signature database. It's created by the people at the Broad Institute, and it has Go terms in it. But it also contains tons of other data sets, and it's a curated gene sets from a variety of sources. You can create, if you have a paper, you can submit your gene sets from that paper to MSigDB. And they are widely used when people do a GSEA. So that's Broad specific gene set enrichment analysis approach. They do more often than not be using MSigDB gene sets. And so we have all these gene sets that we just talked about. We want to use them in R. So how do we do that? Well, a lot of them just exist in R in databases. And so we have these three packages, go.db. This is a Go term database. We have reactome.db. That has a reactome. And we have a GSEA base, which doesn't actually contain anything, but it has tools for manipulating the most common formatted of file for MSigDB objects. You also have the org.db packages, which I showed you 20 minutes ago how to use these to get Go terms. And they're the Bioconductor specific package. And so actually when I'm working with Go terms, I don't actually use go.db very often. I predominantly use org.db. But you can access these in very much a similar way to what we talked about before with using select, and you can dig in and say, I have this Go term, I want to extract out all the IDs, or I have this gene, I want to know what Go terms it's in, things like that. So those are kind of the gene sets, and those are tons of options. I want to focus on MSigDB, okay? And that's mostly because we've kind of touched on Go terms, and people generally know what they are, and doing analysis with them is very straightforward, I think, and we'll show you how to do that. But we just want to be a bit more expansive about MSigDB because they're a very rich source of information and of gene sets specifically. So MSigDB contains a lot of different gene sets. It's predominantly focused on human, but they have these different categories. Okay, so you have hallmark gene sets, positional gene sets, curated gene sets, motif gene sets, computational gene sets. It has the Go terms. And then you also have these oncogenic and immunologic gene sets as well. So you have all these different categories, and you're able to focus on specific subtypes of gene sets, depending on what you are interested in. And you can go to, let's look at the website. You can go to the website and you can download these super easy, and you can get more information about them. So here we are on the GSEA website. It tells you some information about how to run GSEA and stuff like that. But we're here for MSigDB. And so if we look at this, you can like search for gene sets. So I have to log in. It's not much of a login. And then though it's focused predominantly on humans, there are some other options. You can filter by a contributor and say, I am interested in polycomb. You can then look at all the gene sets associated with polycomb, and then often these are related to a specific paper. So you can then look at the paper that's associated with them. And so if you go onto this website, you can download these gene sets, and they typically come in a GMT format. So this is called a gene matrix transposed. It's a GSEA specific format, and it's easy to edit in spreadsheet programs if you need to manually curate it in some way. It's basically tab separated. And so GSEA base allows you to work with these data sets super easily. So in this case, I downloaded the hallmarks gene set. I downloaded all of the hallmarks. So if you remember, hallmarks is this H set. And it's actually, like it says, it's an aggregation of many different gene sets here. So it aggregates a lot of this information together. And it has a lot of things covered within this. So it's a very, I'm struggling for the word. It's a comprehensive, yeah. We got a question a couple minutes ago from Charles. Yes, they do have mouse gene sets. I think something I'll show you in a little bit is also even more useful for this as well. So yeah, you can use this function, git GMT. Once you've downloaded it, this version is also available in the data folder if you wanted to just play with it. But I'm not sure what version they're on now, so this might be out of date. But if you do git GMT, you can read it in. And it's a specific format called a gene set collection object. And so this is basically in a list style. And so what we mean by a list style is it's kind of an object that doesn't have a specific dimensions associated with it. It has lots of different containers for different pieces of information. And so if we looked at the first slot within it, using the double brackets, we can see this contains a gene set, a single gene set. And this is for TNF alpha signaling via NFKB. And so it has 200 genes. Yeah. So you can extract out the specific gene sets, or you can also use the names accessor, and you get the names for all of the different gene sets associated with this. You can also use the gene IDs function. And what that does is it extracts out the actual the gene IDs themselves. And so in this case, it's returning symbols. Okay. So you can get both your gene set name and your gene IDs from this in a really straightforward way. And so we introduced this idea of using GSE base. And because a lot of the time, if you start working with someone and they're using MSigDB, they'll have GMT files, okay? And GMT files, like you can exchange them. They may have created their own gene sets and then put them into a GMT file. It's a relatively common file format. But recently within R, we've kind of switched over to using a new package, which makes life even easier. Okay, and so we don't have to worry about downloading something from somewhere else. We can just have a package that takes care of everything for us. And so this is the MSigDBR package. And if I look at MSigDBR, I use the MSigDBR function within that package. I tell it what species. And so this will get you all the mouse gene sets. And you can tell it what category. And so something I want to mention now that we've got to this point, and I think, you can actually, there's lots of different species associated with this. Most of the species that aren't human, most of the gene sets are derived from just doing like a ortholog analysis. So it's a gene set that exists in humans. And they're like, well, the orthologs are this in mouse, or this in fly, or this in pig. And they will then say that these gene sets are equivalent. So it isn't like there isn't always a one-to-one between the study and the gene set in your organism. Okay, so it isn't perfect, but I've used it for a wide variety of species, and it does a pretty good job for me. So depending on the species, the level of directly curated from that specific species varies. But I think for mouse, especially, you're like fine, because there's lots of mouse studies. Okay. But so when you do this, which database would you suggest for a non-model organism? I think it would depend on your organism. I would say, oh, like ants. Okay, yeah. So typically, I'd say for non-model organisms, what happens is there's normally a consortium. I cannot remember if there is one for ants specifically, but normally there's a consortium of people that work in ants who curate Go terms to help with that. Then, if failing that, there are tools which help do ortholog analysis over a longer distances to then do Go term analysis. So these would include things like Blast2Go and Eggnog. Okay, and so those are much more complicated bioinformatics analysis tools, and kind of get beyond the kind of purview of the very standard analysis with model organisms, unfortunately. But we have done it as a group, but we find it challenging. So that's something that maybe you want to chat with us about if you're doing that. Yes, and then yeah, but you can always just, if you do have reasonable amount of orthologs, and you're able to compare it to something like Drosophila, for example, for ants, then you can start to get some intuition. It might not be perfect. You might not get there are different molting behaviors and things like that, right? So it might not be quite perfect, but the big issue is really just getting the orthology between your gene IDs for ant and your gene IDs for whatever closest organism that you can use that has functional gene sets. So yeah, sorry that isn't an easy answer for that, but it does vary quite a lot. Yeah, some non-model organisms have a very good, well-established Go terms and things like that, but others less so. Cool. So when you use this msigdbr function, what it provides, instead of this GSEA collection or anything like that, it just gives you a specialized data frame called a table even. And this is basically a normal data frame. It's just a very specialized kind. And within it, you have things like the gene set category. You have the name of the gene set, gene symbols. There's a row for every single gene symbol. The gene ID, the ensemble gene ID, the human gene symbol. And so it's really nice because it has all this information that applies regardless of what your data set's in. So maybe I did all my analysis and I was using ensemble genes. Or hey, this has the conversion and it will tell you which gene sets they belong to. Yeah, so this is a really nice resource for this. And so once you have, we've shown you these gene sets, and we've shown you how to where they are, what packages they are, how to maybe access them or read them into R. Next step is to actually do a test. And so the first method that we'll talk about is functional enrichment, okay? And so this is really the idea of I have X amount of differentially expressed genes. Say I have 100 differentially expressed genes. And I have a gene set that has 20 genes in it, and actually 15 of those 20 are also differentially expressed. So that would suggest that if I have 100 differentially expressed in total, and it would suggest that that's quite a large proportion of that gene set which are included in these differentially expressed genes. And so it's essentially performing a simple Fisher test of I consider all my genes, I consider my differentially expressed genes, I consider all my gene sets, and I consider how many differentially expressed genes are in that gene set. GSA works a little bit differently than that. And so that's our second method. And so this instead ranks everything from top to bottom. So you're not just focusing on differentially expressed genes. You're not just focused on a cluster. Instead, you're focused on a ranked list. And so this could be my log2 fold change that has been shrunk, and it's been ordered by that. And then it sees, is there an association with my gene set in this ranking? Is my gene set all at the top of the ranking or at the bottom of the ranking? And we'll get more into the specifics of that in a sec. But first, we'll start with just working with doing a functional enrichment style test. And so to do this, we'll need some data. So we can read in this CSV file here. And so this contains log2 fold change information. It's the result of DE-seq2. And you can see here's the first 3 columns. We've still got the NAs in here. But it's essentially the result of our activated versus naive cells. We also have the ID, and we also have simple information. So we have lots of information within this. Sorry. So first, before we can do anything, we have to do the NA filtering. Okay, so this is something, a repeating step that we're kind of learning about as we get these NAs. Often, if you supply a value with an NA to a tool, a package, it will just break because it's not expecting NAs, it's expecting numbers. And we also want to increase the power. Okay, so we increase the power by taking out genes that are not interesting. And so this NA allows us to filter those genes out, and so we can filter it down here. And so now you can see we filtered out these genes, and the top of our list now has some interesting candidates like this one, which is very, very significant, which is MTN1. So now we have our data, we have our genes of interest, we can actually start to do our functional enrichment. And so we're going to tell you about 2 packages to do this. And one is very comprehensive and it can do everything. Okay, but it is a little underpowered in a very specific way. And so that's what we'll talk about second. But first, we're going to talk about this package called GoSeq. So GoSeq is really great, but it isn't quite as comprehensive, do it all, as the other package we'll talk about in a sec. And so what you can do with GoSeq is you provide it with a name vector of 1s and 0s, and the names correspond to genes, and the 1 or the 0 correspond to whether it's a gene of interest. Okay, and so that could be it's an upregulated, significantly upregulated gene, and you want to do a test on significantly upregulated gene, so it has a 1. And so to generate this list, you can just take your adjusted p-values. We say, is it less than 0.05? And then we can also get the log2 fold change, and we say it's greater than 0. Okay, and so what we're asking here is this gene significantly upregulated? We then convert it to an integer, because the product of this is true, false. If you convert it to an integer, it will be 1, 0, and 1s will be true, 0s will be false. And then you add the names. And so the names will just be the IDs. And so if we look at the first 4 of these, all of these are false, and they have their names here. And if we do table on this object, what table will do is it will count how many 0s there are and how many 1s there are. And you can see that we have nearly 2,500 genes that we're actually asking, are these genes enriched in anything special? Versus a background of 10,000. And so then next we're going to work with GoSeq, and GoSeq has a function called supported genomes. So it has a wide variety of genomes that you can use with this. So potentially, I haven't checked, maybe they'll have some ants in here, and in data. Maybe they won't, but they might have something close enough. But yeah, here we got a lizard in here, so it's not just model organisms. And so you can check which supported genomes are available and which database they're from. And so this is just a mouse data set, so we're just going to be using MM10. So it's nice and straightforward. And so this is the strength in GoSeq. Sometimes you can have length bias. So say you have some length bias in your data set, and there's also there are length biases in Go terms, right? Some, like for example, essential genes are quite often a little bit shorter because they need to be expressed more. It allows for transcription to be faster. They're often shorter, they have less introns because they need to be processed and delivered quicker. And so if you have any length bias within your data set, and this could be technical, it might not be, but it can bias the results of your GoTerm analysis if you have length bias in what you're testing against and length bias in your experimental data. And so what GoSeq does to address this is it does a null hypothesis, and it does a model to assess what the relationship between length and your data set is. So first, we'll run this null p function to get this kind of length-based model. And so the important thing with this is by providing MM10, it can know everything. It can access all the database information about this gene. So we say we have this up in ACT. If you remember, this is this guy. Okay, it has the gene IDs. It can then look up. You tell it it's MM10, so it knows the gene IDs. It knows what genome it came from. You tell it you're using known gene tables. So this means it's going to look at the known gene tables from UCSC. This is what annotation it's using. Okay. So this means it's like the GTF or the SAF. And it's going to have a look at those to kind of infer length information about your gene. And then you can ask it if you want to get a plot. And then when you get something back that looks like this. And so you can see that there's actually, if there was no bias, this would be straight. But this is quite bent. And so this would suggest there's actually a length bias in your data set. And so then when it comes to doing GoTerm analysis, it accounts for this length bias. So if you have a GoTerm that has lots of short genes within it, it will downweight it. While if you have a GoTerm that has lots of long genes, it might upweight it. And so once you performed that analysis, okay, so right, we saved this null p as PWF. We can then provide that to GoSeq, the GoSeq function. And in this case, we're going to test it with GoTerm. Specifically, we want to look at the biological process. And so there's a variety of categories available for GoSeq. It has all the GoTerm ones, and then it also has KEGG. So it doesn't have MSigDBR or MSigDB even. So that's unfortunate because that's a super useful resource. And so this is maybe one of the drawbacks of GoSeq. But we do this analysis, and then we get the categories back, the p-value. Is it overrepresented? Is it underrepresented? So it's doing both tails of the comparison. The number that exists within the category, and what's the term called. And so unsurprisingly for this T-cell analysis, we're seeing immune system processes come up, defense response, response to stimulus. Okay, they're all super significant. So this suggests it works, and GoSeq, I think it's always a nice like first port of call, especially like you can do this kind of gene set analysis for like if you're doing ATAC-seq, you're doing ChIP-seq, you're doing all these kind of other any kind of enrichment analysis if you have a specific group of genes that you're interested in. But especially when you have RNA-seq and you have this potential for a length bias because of the differential amount of the way that RNA-seq libraries are prepared, it's good to always check this to see what your null p is doing and to see if there's maybe a model I need to take care of. Okay, and so once you know you have a GoTerm that's interesting, so like immune system process, you can then go back to things like the org and then EGDB, and you can extract out the Go information. You can extract out the entree IDs. And so in this case, I'm going to this org and then EGDB. I'm saying I have a key type, and so I have a Go term. So I'm saying Go all, and then I'm providing it with this Go term. And I'm telling it what I want to get back is gene IDs. And so this is for specifically the immune response group. And so then I can extract out all the immune response genes. And then I can compare the immune response genes to the results of my differential gene expression analysis. And I can see what are the specific genes that are driving this result. So I get all the IDs from here. I'm making them unique because sometimes you get duplicates because they might have come from different sources. And then I'm asking which ones in my analysis are from this gene set. Okay, and then you're able to see, you're able to go from your differential gene expression analysis, you find out which Go terms are enriched, then you can actually look back at your differential gene expression analysis at those specific Go terms, and then you can do things like make heat maps like we did in the last session of these Go terms. That's quite a common next step, is you'll take this gene set, maybe you go back to the R-log counts, and then you'll extract out those information and you do a nice heat map. Or you'll look at what are the log2 fold change values of this gene set versus several other gene sets. So you can see the amplitude of change. Okay, so that is GoSeq, and we really like GoSeq, and the real big strength of it is this kind of length bias. Okay, it has this ability to help you deal with technical artifacts in your data set, which a lot of the time is not a big deal, but sometimes it can be super problematic. Okay, yeah, so we got a question with about is GoSeq analysis the same as the David Go analysis? So I'll have to double check. I believe that under the hood, it's the same significance test. However, I don't think David is accounting for this length bias. I could be wrong. I haven't used David in a long time. But I think one of the things I want to mention, and this gets back to being a good bioinformatician and being a good data scientist, essentially. Often people use David through a web browser, right? So we could go, should we go to David? Oh, I forget that my internet has been terrible. So yeah, we can start doing an analysis. I can paste a list. I can do stuff through here. This is, I'd say, is most of the time how people do this. But when people do this, they don't save the version of David they're using. They don't keep a track of exactly what gene list they're using and what version. They don't write down the date. But when you're doing it in R, you installed a package on a specific date. You're using a very specific gene set. You can, when you do your analysis, you can save this session information from your R session. And so when you're finished, you can reproduce everything you're doing super easily. And so this means that if you come back 6 months from now and you run it again and you get a different result, you can explain why. And it is relative, like especially with Go term analysis, we often get this where someone comes in and they're like, it's not consistent results. And it's because they're using like a web browser and it changes over time. The tool changes over time. And maybe it's reordered, and maybe their favorite term has dropped from 1st position to 6th position. And it's not the biggest deal in the world, but it could be avoided if you're using a consistent tool. So yeah, if anyone like is interested in good coding practice and things like that, I'd also point you to another thing that we have. So we have a section called reproducible R. It is in, here. And this tells you how to use, to make reproducible reports in R using R Markdown. And this is really important because then you can contain all the session information and you tie the output of your results to the code that you use to generate. In fact, these slides are built in R Markdown. So you can see that I have code and then I have the output of the code and they're next to each other. And so you can create like a nice report with your analysis and to help tie all these things together. So that's our view on it. Under the hood, it's going to do something very similar. It's going to do a functional enrichment. But it just has limitations. Okay. Oh, what do I think of ingenuity pathway analysis? Yeah, it's great. So one of the things that I haven't touched on about this and specifically relating to gene sets is some of these things are proprietary. And so we used to show people a lot more in depth how to use KEGG, but KEGG moved behind a paywall. And so some people have access to parts of the KEGG, and some tools still use KEGG, but actually getting all the information for KEGG is harder. And similarly with ingenuity pathway analysis, I've never used it because it's behind a paywall and it's not necessary for what we do on a day-to-day basis, because we have other tools which are free to use and available to everyone. So we know of many labs that like using it and like it, and we just don't see it personally as necessary when we have other free tools that don't cost scientists money. We like open source software where we can use it. That said, it's supposed to be a very, very nice curated data set. So there's a reason that it costs money, because they've put a lot of effort into it. Yeah, but I'd be curious to say, like, once you've learned how to do this, compare some of the results and see if it's fairly consistent, and like what is ingenuity adding to your analysis. Is it adding anything? Because if it is, then it's 100% worth it, right? But if it isn't, then maybe there's other tools you can use. And I think ingenuity, I believe, is more like metabolomic focused. So you might want to focus on things like the reactome.db and things like that, or maybe using some of the tools that allow you access to KEGG without having to sign up for KEGG. Okay, so yeah, I showed you how to do this filtration. We're moving on to cluster profiler. And so as great as GoSeq is, I love cluster profiler so much more because it has everything. And it's a very, very, very comprehensive tool set. And it allows you to do so much in a very quick and easy way. And most importantly, it makes some very nice plots very quickly and easily. Okay, so I'm going to give you a quick run through, but I would say that there is a, I think this is linked to it, there is a very large book that tells you everything that you can do with this this package, and it's fantastic. The guy who does this, he does a lot of bioinformatics development, and he's very, very present within Bioconductor and the R bioinformatics sphere. So he's a really, really good developer. But yeah, so Cluster Profiler allows you to use lots of different functions in the same way. Same common way of using it, but you can do it for Go, you can do it for KEGG, you can do it for MSigDB, but it's a common way of doing it, which is nice. And as I mentioned, super nice visualization. And so if we're going to just do this for the kind of same approach that we've been doing, this gene set enrichment, this functional enrichment, there is a package, a function called enrich_go. Okay, and so this is still for Go terms. There are other ones which are less specific, and there's ones that are specialized in other ways. So for example, there's enrich_KEGG, which is for KEGG terms, and there's enrich_R, which allows you to provide user-provided gene sets. Okay, and so you can use enrich_R with MSigDB, for example. And so the inputs it wants for this is it doesn't need any ones or zeros or anything like that. It just wants a list of IDs. Okay, so in this, we're saying, I just want to get the IDs that are significant. I'm not looking for upregulated or downregulated. I'm just looking for significant. And I'm extracting out the first column because that's where the IDs are. And so I have this vector of IDs. And so then I provide this to enrich_go, and then I also tell it which org-DB it's from. Okay, and so this is important because then it knows what the universe is. So it knows what gene, to be able to do this kind of enrichment analysis, you need to know which genes are significant out of what universe of what are all of the genes. Okay, and so when you do this, if you just provide org_mm.eg.db, it will then just assume all genes were potentially going to be differentially expressed, and that's your background. You can customize this universe though if you want, and sometimes that's really important. So with as simple as doing enrich_go, you get your enrichment result. And within this, it's a special object. Again, it contains lots of information in it. It's an overrepresentation test. And you can already tell there's some information if you just look at it. There's 493 enriched terms. It starts to describe what they are: ubiquitin protein ligase binding, actin binding, etc. But then it cuts it off because it doesn't want to show the whole thing, saving you a bit of memory. You can just take this object and go as.dataframe, and it will turn it into a nice data frame that you can look at and or export using like write.csv or Rio export functions and put it into an Excel spreadsheet. But I think the really exciting stuff with what we're doing here is you can then do some of these visualization tools. So here we just called the dot plot function from Cluster Profiler. We do it on this enrichment object. In this case, I'm saying I want to see 6 categories. And I'm also doing it's a ggplot object, so you can customize everything. So in this case, I'm making the text size a little bit smaller because some of these terms are super long. And so you can see here, it's a dot plot. It has on the x-axis the gene ratio. So that's the proportion of the gene set that are significantly changed. Okay, so out of the ones that you gave it, what proportion of the gene set in question are these significant genes? You have count information for what is the number of significant genes that corresponds to the size of the dot. And then you have the significance score. So the darker the red, the more significant. And so it's organized by significance. No, it's organized by this gene ratio even. And so you can see that we're getting some transcription. These are quite some big terms. you have actin binding, transcription factor binding, and so kinase activity. So these are some quite large terms. You can also do these kind of nice plots. I need to make this smaller, but I can't now. So you can create these things called enrichment maps. And these are network plots, and they show you how significant groups in the gene sets relate to each other. So often gene sets that you find are significant actually have a lot of overlapping genes. And so you might actually get, like, say you get 20 different significant genes or gene sets. You might find that there's actually just 3 or 4 clusters of gene sets which are really related to each other, and those are what's driving it forward. In this case, it's not super like that, but you can see that we have, like, what the dots are showing are these individual gene sets, and the lines and the thickness of the line corresponds to how many genes are overlapping between them. And so you can see that actin binding and actin filament binding gene sets are kind of related to each other. Transcription co-regular activity and DNA binding transcription factor binding are related to each other. And so this can be a nice way to, like, parse out, like, themes within your Go terms, depending on what the results look like. This one, it's a little bit sparse, but most of the time you get, like, a lot of related terms to each other. Okay. So the last thing we'll cover is GSEA. And so all of the Go term analysis and gene set analysis we've covered so far is really focused on this functional enrichment. Do you see more in your group than you expected by chance? Okay, and it's a simple, just like overlap analysis and enrichment analysis. GSEA is much more complicated than that, but the nice thing about it is it allows you to leverage your whole data set. So doing a gene set analysis, the previous set relies on you having a cutoff, a threshold of some kind. So in that case, we're using an adjusted p-value of 0.05. But there could be lots of useful gene set information at 0.06, right? There could be extra things there that may have, like, pushed it over if we did it for whatever value, for whatever reason we were considering 0.06. Or maybe a more significant, more stringent threshold would have been more useful. And so by using all of your data as a ranked list, it then allows you to use everything, and it's not about cutoffs. And so the way that this works is you have your ranked list, okay, on this XY. And every time it does this random walk, okay? So it will start at this left-hand side, and there is this 0 value here, this black line. And then every time it hits a gene that is part of the group it's testing, it will go up. And every time it hits a gene that's part of the group that isn't part of the group you're testing, it goes down. Okay, and the rate it goes up and the rate it goes down corresponds to the rates, the overall proportions of these gene sets. Okay, and so if the gene sets, say we were looking at immune system genes, if they were randomly distributed throughout your ranked list, then it would go up, and then it would go down, and it would go up and down, and it would stay pretty much at the 0 line. Okay, because it's kind of randomly distributed. But if they're all up here in this significant region, if they're all at the top driving, say they're all upregulated, then it would go up, and then it would go up, and then maybe it would go down occasionally, but it would go up and it would keep going up. And then eventually it will drop down. Okay, and that's exactly what you can see here. And so by doing this approach, you can then start to leverage, like I said, everything in your data set. But you can see why it's important to do the LFC shrinkage, because you don't want this genes that are ranked high to not be ones to be ones that aren't significant. You want to downweight the ones that are not significant because they probably have a big log fold change that are just through chance. And you want the ones you're confident in to be at the top of your list. Yeah, so that's the idea. It's called a modified KS test. This I think is I can't remember is two people's names. But if you go to the Broad's website, they can explain. There's various walkthroughs that explains the theory behind it in detail. I think the big thing to take from this is where it hits its peak. This is considered to be the enrichment score. Okay, and every gene that is in your gene set of interest that is before that enrichment score is considered to be the leading edge. Okay, and so we have the quick question. I think you kind of got it. Is it ranked by fold change? It can be. And so if you're going to do fold change, we very much recommend it's the shrunk LFC value. However, we quite often use what was used in the original paper, which is the test statistic. Okay, so in the DE-seq2 column, this is stat column. Okay, some people also use the adjusted p-value as well. So there isn't one specific right way to do it. The original paper uses the statistic score. I'd say more people than not these days use the log2 fold change. But if you would do that, you've got to make sure it's shrunk first. Okay. And I just saw the question from David. For the Go analysis, is there any benefit to analyze one heat map cluster at a time, or would you pass in whole differential genes? Yeah, so if you do clustering analysis, definitely what I end up doing is I do a bit of everything. So I look at the differential gene expression analysis, and then I do some gene set enrichment analysis. But then I'll also do some clustering, and then I'll check each of the clusters and see if any of the clusters are enriched in a specific gene set as well. So I do a bit of everything for that. Yeah. And the nice thing about this is you can also, there are tools, I didn't get into it, but there is a function within Cluster Profiler called theme comparison, and you can run gene set analysis on multiple groups at once. So you could do all of your groups, like say we had our data set from earlier where we had 7 clusters, we could pass all 7 clusters of genes at once to the gene set analysis. And when we do this, it will show me the result of all 7 at once relative to each other. So you get an understanding of how they are related to each other. Oh yeah, was TopGo? Yeah, we like TopGo. We like TopGo a lot. We just find TopGo is really nice because the way it does the Go analysis, specifically for Go term analysis, it builds all of the trees itself, and so it has a lot more information contained within it. You can also do more complicated algorithms for deciding the enrichment analysis. So when we do the enrichment analysis, it does the very basic enrichment score. But quite often we actually prefer, there are some other algorithms like I think it's called parent-child algorithm. And so that statistical test instead will downweight some of these really big terms and promote some of the smaller terms. And so by downweighting some of the big terms and promoting some of the small terms, you're actually able to get some more specific results from your Go analysis. So we do like TopGo. It's just, it's a little bit like cumbersome, and it is quite slow because it is quite like what it's doing is quite intense. But yes, we do like TopGo a lot. And then a follow-up to the cluster question, but do you pull all clustered together or pass in as individual groups? So the way that it works so if you just put everything together in into one analysis, like just all together, then it would just be your entire experiment, right? And so you wouldn't be able to look for enrichment in a specific group. But what you can do is create a list, and in each slot in the list is each cluster. And then you supply that to Cluster Profiler using the theme comparison function, and it will then be able to like pass all 7 and do it all at once. It's really nice. But yeah, look in the textbook, the online book they have, and it will walk you through how to do it. Okay, so yeah, so this is a kind of a question we just explained a few minutes ago. Here you could use the log2 fold change if you've done LFC shrink, but we're going to use the stat column because we like the stat column. Okay, so we're creating our ranking. And so we take the stat column, we're taking out specifically the stat column, we're then giving it names. So this means it's a vector, and it has the value of the stat column and the ID associated with it. And then we're just ordering, and we're doing it in a decreasing value. So it starts with the highest values and it goes to the lowest values. Okay, and so that's all you need as your input for GSEA. And so we're going to do this using Cluster Profiler, and we're going to use the msigdbr package. Okay, so we introduced GMT files and said there are other packages that do GSEA, like FGSEA. But while we're using Cluster Profiler, it actually, all you need is a simple data frame. So it works really nice with the msigdbr package. And so we're just going to look at, because we have these immune cells, we're going to look at the immunological signature group. And so Cluster Profiler, all it needs is a 2-column data frame with the gene set names and the gene IDs. So we'll do msigdbr on mouse to extract out C7, and then we'll subset it down to just the GS name and the entree genes. Okay, and so that's the product here. Okay, so we have the gene set and an entree gene ID for what's in it. And then we just run the GSEA function. And so this is the ranked list. This is the term to gene, so that's what we just created. And this is a cutoff. So above a certain value, it will stop reporting higher significance scores. So I like to sometimes put in a really nice value here, so your most significant scores aren't all just exactly the same thing. And so you can then just do the same functions on this enrichment result. So this thing here is exactly the same as what we got before. It's this object, okay? But it's just had a different test, and it's had the GSEA test. And so we can still do dot plot on it. And so here you can see what terms we're getting out. We get a lymphoid progenitor cell, something about T cells, another T cell set, another T cell set, another T cell set. And you can see they're actually quite different from each other. And normally the first name within an MCDB object corresponds to the study. And so you can see they're all from different studies. And so you can then go back and look up this on MCDB. You can go and look at the study. You can see what the overlaps between this study is and your study, and start to learn a lot more about what's going on. We can then also draw these plots, these maps, and you can see how these are related to each other now. And so you can see that this one, the skin one, is actually completely different. And so you have all these T-cell related gene sets, and then you have this skin one, which actually has a different overlaps. So this one might actually be really worth following up, because it might contain some information that is maybe not as expected, or it might be something that hasn't been characterized to be T-cell before, or it might be something that's to do with an artifact. Okay. And so Wuji has a question. Do you have any suggestions to deal with genes with the same rank? Okay, so it happens occasionally that your statistic score or whatever score might have exactly the same value. It's rare, but it does happen. There are control within GSEA function, I believe. Let's see. Yeah. Just share with you real quick. Okay, so if we go GSEA, does it have an option here? Okay, it might just not be included in this help, but I know that they have specific instructions for how to deal with matches, and so you have there is a built-in option. I just don't know how to get to the option. I can't remember off the top of my head, but within Cluster Profiler, I'm pretty sure there's a built-in option for what it should do, and it can just do it randomly, essentially, over an entire gene set, like a large gene set, a large experiment even, sorry, a large experiment. Ties are not going to make too much difference in a GSEA unless you have a lot of ties, okay? And sometimes your data can stratify for some reason and you get a lot of ties, but generally it won't. If you have a lot of this stratification, then there's probably something up with your data, and you might need to do an alternate approach. You can, at that point, if you have a lot of ties in your statistic, maybe try doing LFC shrink. That might be a way around it. But if it's just a couple ties here and there, it's not too big a deal, whether it does it by just randomly assorting them. It shouldn't affect things. Okay, and so then the last plot is this running score plot. And so this is a very, very typical GSEA plot. It was on my intro to GSEA slide, and it's very easy to produce these, and it's just GSEA plot, okay? And you provide it with an ID, okay? So in this case, I just provide it with the ID. I didn't actually provide the ID, I just said I want the first one, okay? And then I can give it a title. But you can see here, these black lines correspond to the gene set we're testing. They correspond to genes within this HSC versus lymphoid. And so you can see they're all top in the, like, very highly ranked. And so this is you can see why this is so strongly overlapped. Okay. And so I think that's us. Thank you to everyone for sticking around. I know it's been pretty intense, and I apologize for overrunning in every single session. Normally I'm pretty good with the time, but I'm obviously a little bit rusty and not talking to enough people. But if you need any help, any follow-up, please feel free to reach out. Remember, we're brc@rockefeller.edu. You can just send us an email. If it's just a couple-minute question about some of the course content, that's totally fine. If you want help with your analysis project, that's also really great, and we'd be super stoked to help out. I will be doing the exercises and running through them in about 20 minutes. So if you do want to stick around and do those, you can. If people have other questions, post them in the chat. I'll be here. And let me think. If you do the exercises at another time, we really recommend, if you need help, going to the Getting Help section and posting questions here. Okay. If you email me directly about exercises for help, I will probably ask you to put it in here anyway, because it's a good reference for us. And it's a good reference for other people, because quite often people can see the answers to questions they already had. And then lastly, if you are interested in differential transcript utilization, this is really great and you should look at it. Okay, but I barely fit it in, everything else in. And so we couldn't have another session. Yeah, I think that's it. I will also email everyone once the YouTube videos are up as well, so you can review them there. And I will also be reaching out, hopefully soon, about a full-length course covering all of our course material, which it's a lot, and you can dip in and out for that as much as you want. Okay, I'm going to make myself scarce for 20 minutes. If anybody needs any help, just post in the chat. Thanks for all the kind words, everyone. Hello. So yeah, there's still a few of you here. I'm just going to run through the exercises real quick for this section. Okay, so we've got the exercises. Okay. So these exercises focus on gene set analysis, which we just covered. But the data sets used in this are actually going to be dipping back into Bing-Wren's data set of liver, heart, and kidney. We're just going to focus on liver versus heart. I think it's mostly just going to focus on the results of differential gene expression analysis, but we did a bunch of clustering and stuff, so you could also use those as well as your inputs. But yeah, so the first question is identify GoTerm cellular component groups enriched in genes significantly upregulated in liver with GoSeq. What are the top 5 terms? So library, I have library cluster profile here, which we will need, but we won't need it quite yet. So let that in. So everything is in the data folder. So the heart minus liver CSV. This is a CSV folder this time. So we can do read.elim to do the CSV. We tell it the separator is a comma because it's a comma separated file. And so we can have a quick look at it. See what this data looks like. And you can see it's the result of DE62. And of course, you can see at the top, NA value, our favorite. So we're going to filter those out. It's .NA. Let's get rid of it. And then we specifically want the genes that are significantly upregulated in liver. So we're going to do up in liver. It's less than 0.05 and less than 0. So in this comparison, when we set it up, liver was the denominator. So it'll be less than 0. Okay, so now we've just asked the question, which ones are up in liver? And we get a bunch of true falses. We need to convert that to an integer. And now it's a bunch of 1s and 0s. And this is what GoSeq likes. So it can't just have 1s and 0s. We also need some IDs associated with it. And so the order is maintained. So we can just directly take the IDs from our original data frame and assign them to names within this vector. So if we now look at this, it's still 0s and 1s, but now we have names associated with each of the 0s and 1s. And so we can do library GoSeq, and then we can do our null p plot. So you can see it go. And so you can see there's definitely some bias there, right? We have this big swing up here. Again, we want it flat ideally. And so we can run up. Now we can just run the GoSeq function. We run it on the PWF. And because I specifically asked for the CC function, right? CC. So we'll do go CC in the test category. Right, so that's going to take a sec. And so if we think we're doing heart minus liver, so these tissues are quite different. And so we can have a guess what the top 5 terms will be, but I think this fits kind of what you would expect. Endoplasmic reticulum, extracellular space, extracellular regions, peroxisome, things like that. And so you imagine there's a lot of specific things related to the liver, like to do with processing and secretion and all that jazz. And so I think that's really what's manifesting here. And so it's going to be, yeah, that's going to be why we see these. So the next is we're going to switch over to doing cluster profiler. And so what we want to do is plot the minus log 10 p-value for the 5 terms as a point plot using the KEGG enrichment. Okay. And so I've added a hint here, GSE KEGG is the cluster profiler function for looking at KEGG terms. So we need our gene list. Okay, because we're doing, hasn't actually, that should be in the question. We want you to do a GSEA with this. Okay, and so if we're going to do a GSEA, let's do stat. Let's get the stat list. And then we have to associate names again with this list. So if we look at gene list now, names and stat values. And then last but not least, we need to organize this. So we have to sort it. So if we look at head gene list now, you can see it's all the highest scores at the top. So now it's super simple. We can just run GSE KEGG. And it's asking for what? It wants your gene list, it wants key type information, the organism, and it runs through. And so it has to get read the KEGG information from online. And so we can have a quick look to see what this looks like. Right, and so actually, let's make this big for a sec. Now that we're working interactively with this data file, I kind of want to show you what it looks like. So if you remember, this is an enrichment result. So if I just do this, you kind of get like this weird abstract summary. It has some useful information. We can see that we have 136 enriched terms, stuff like that. Super good, really interesting, useful stuff. But often we want to look at it as a data frame. So if you just do head, which gives you the top 6 terms, it'll show you this. And so you can see description, set size. Look at these terms coming up. This makes sense, right? For liver and comparing liver to heart, we have some cardiac, cardiomyopathy, but then we also have oxidative phosphorylation, retinol metabolism, like muscle versus liver explains the difference. And so then we also have things like the enrichment score and the normalized enrichment score. These are these two. If you remember, these are relative to the highest point in the peak of deviation when you calculate the KS metric. Adjusted p-value is super important, rank. And then the core enrichment is the thing I wanted to get to at the end. Okay, so this is the final column, and it is a list of gene IDs separated by a // And so you can extract these out of here, and these correspond to the leading edge. These are the genes in the leading edge. These are the genes driving your enrichment. So I just thought I'd point that out while we were doing this. And there's lots of cool information in this object. Okay, but we have the top 5. I think they all make sense. Some of them are like muscly, and some of them are like more liver. And so then we can do a dot plot. And this one I'm doing big. I want to see 20 terms. So again, a lot of these make sense. Retinol metabolism. Chemical carcinogenesis, I'm not sure. Maybe this makes sense. I don't know. Myopathy, cardiac muscle, metabolism, cardiomyopathy, like, yeah. And so that's your dot plot, and then we go to enrichment plot. Okay, so something I didn't know when we did this in the actual course content, this relies on another related package called Enrichplot. This is made by the same person, and he just separated out some of the functions into 2 plots. And so first you have to use this function called pairwise termism. And so this allows the gene sets to kind of be related to each other and to build up this network. And then you can actually run the EMAP plot. So we have it here, but you can see what I mean by like using these maps. It can yield some really interesting results. You can see how we have this oxidative phosphorylation. I'm assuming this is all related to, this is a liver specific. Then we have some cardiac specific related to cardiomyocytes and like calcium signaling, so there's like muscle contraction a little bit. And then we have this separate group, which is related to drug metabolism in general, and steroid hormone biosynthesis, retinol metabolism. So I'm guessing this is another set of, this is kind of your liver signature. Sorry, this is your heart signature because it's oxidative phosphorylation. So it's like, you know what am I trying to say? It's to do with muscle contraction, it's energy consumption, it's respiration, right? Then you have a different heart signature here that isn't as much related to respiration. This is more to do with the actual contraction. And then we have a few loose categories here. So that's why we really like using these maps, because it can actually help understand how these terms are related to each other and interact with each other. Okay, heat map of the leading edge of the genes. draw a heat map of the leading edge of the genes driving enrichment in the top 3 code nodes. I think I actually just did the top 1 in the example. Okay, so you could just do top 1. And so first off, we're going to have to ID and identify and extract out these gene IDs. So this is from the core enrichment column. That's what I was talking about earlier. So we're going to need our p heat map. We're going to need DEC2. We're going to load in our gene IDs. And so what we're doing here is we take column 1. We take the first value from column 1 of the core enrichment column, and we're splitting it on the /. And so it's taking something like this, and it's splitting it down on the /, and so that we have all of the gene IDs as separate entries in a vector. So you can see that here. So now we have all of the gene IDs. We are ready to subset, okay? But if we're going to be drawing a heat map, we're going to need counts. And if we want to do counts, we're going to have to load in the count data. So we'll load in the count data here. And then we're going to have to do rlog. So I'm going to just set up the DDS object. I'm going to run DEseq. And then we're going to do rlog. Right, so now we're going to have our transformed counts that are ready to go into input. We need to subset them. So we can use the in operator to find out which ones of our row names are in the gene IDs. I don't think I've shown this broken down yet. If we do that, because there's true falses. And so we can then index this row from the rlog tissue based on whether it's truly in that list or not. Okay, so I didn't actually save that somewhere, but you can see this is our full count table. And so if I save that as our matrix for the heat map, it's ready to go. And then the last thing we'll need is annotation data. So we can grab anno.df. So this is our col data. And again, we'll drop the size factor. So we'll do drop equals false because we don't want it to turn into a vector, remember? So now if we look at that, it tries to make our life easier for us, but it actually makes life more annoying. And so we do drop equals false, and then it stays as a data frame. And this is the format that pheatmap likes. And then we get our result. And so it's unsurprising. We've included everything in here because I often think it's nice, even if you're doing a pairwise comparison. If you have a larger data set, it's often nice to see what's going on in the other one as well. And what we can see here is, yeah, unsurprisingly, the genes that are upregulated in the heart are upregulated in the heart. But it's good to confirm that for your gene set, right? It's good to be able to also show, say, we see enrichment of these genes, and this is why. Now you can actually see all of these genes together. And so often this is a nice counterpart to a gene set analysis for the few gene sets you really care about. And it's interesting, there's one set that's actually more significant to kidney. Just a few in the middle. But yeah, so that is it. That is the exercises. So we're all set. I am going to stop recording in a sec, but just say if you need to mention again, if you need to reach out for help, you can email us if you need help with the analysis in general, or you can click on the getting help here if you need help with the exercises. Great. Well, thanks for joining us for the course.